All right. Yeah, so thank you all for being here today. I'm going to talk a bit about some of the work I did during my PhD here at Lund, working on how agricultural intensification has affected genetic diversity in grassland butterflies. And this project sort of is done in a context that I think many of us are very familiar with, that biodiversity is declining globally. And one of the things that we have seen increasing news about in the past 5 to 10 years is the sort of scale of global insect decline, where we're seeing these massive losses of insect biomass and species diversity globally, and that these trends tend to be even stronger in, say, Europe and North America. And these declines in insect abundance and diversity are for the same reasons as pretty much all of biodiversity declines, the same sort of culprits. Climate change and changes in land use and configuration are seen as some of the biggest components driving this loss. And in Europe in particular, one of the habitat types that's seen considerable loss over the past century is semi-natural grassland habitat. And for a sort of temperate continent that globally is relatively lacking in biodiversity, these semi-natural grassland habitats are some of the most biodiverse on the continent. But they've generally been lost considerably due to changes in agricultural practices. They've been either abandoned or converted into large-scale sort of arable land for growing large monocultures. And where I work in Sweden in particular, over the past 150 years, we've seen a sort of greater than 70% reduction in the area of this grassland habitat. And this has had dramatic effects on grassland species that live within them. And so this data isn't from Sweden, but it sort of shows the trend that we're seeing all over, that generally we're seeing declines in insects, but that these declines are even greater in more specialized insects that have specific needs for habitats and for host plants. And so we're seeing this in Sweden as well. We see these loss of both habitat generalists and habitat specialists, but what remains continually shifts more towards more generalized species. And while a lot of the work that has been done looking into biodiversity decline has focused on ecosystem and species diversity, only recently have we really been diving more into how these declines are affecting genetic diversity. And this is important because biodiversity as a whole is composed of these three sort of levels of biodiversity, and they are tightly interlinked. So changes in one cascade to the others. And so loss of genetic diversity reduces adaptive potential and can cause the accumulation of deleterious mutations that drive populations further into decline. And then this sort of cascades up to reducing species diversity and eventually ecosystem diversity. So maintaining all three of these levels have become a major goal for the global post-2020 global biodiversity framework. The goal is to try to maintain these levels of diversity in the next 30, 40 years. And looking into genetic diversity is something that's becoming increasingly available for non-model organisms every day. So this is an example for insects, but over the past 5 years, we've gone from hardly like dozens of chromosome level insect reference genomes to over 2,000. And now we've just hit 1,000 Lepidoptera chromosome level genomes. And so it's becoming increasingly possible for species of interest to be examined in population genetic ways, so we can understand how biodiversity decline is affecting how it's affecting genetic diversity. But assessing this can be quite challenging, largely because there's quite a variety of quite a lot of variation in sort of absolute levels of genetic diversity across taxa. And it's been pointed out many times that genetic diversity levels don't necessarily correlate with conservation or threat status. And this is because they are driven by factors that are occurring over hundreds of thousands to millions of years rather than recent demographic effects. So here, as an example, I have from Macintosh 2019. This the is looking into different butterfly species. We see that their genetic diversity can vary considerably across taxa, um, and that it's not necessarily tied to their phylogenetic relatedness. And for instance, in butterflies, it tends to be related to things like body size, and that has to do with sort of long-term density limitations for population sizes. And so if this we can't have some sort of estimate of this level of genetic diversity is needed for a healthy population, then a better way to look at it is how the sort of trajectory of the diversity for each population. And this is done through genetic monitoring, sampling at multiple time points. But then, unfortunately, that means that we will miss out on genetic diversity decline that has already occurred, or it may take a considerable amount of time for us to notice changes, as often these changes will lag behind the actual demographic decline. And this is where museums can provide a valuable resource. We can, using museum specimens, assemble whole genomes from past time periods, and use these to sort of get a direct estimate of different genomic properties in the past, before, or early on in a disturbance event, and then compare them to contemporary sample genomes and see what has changed over the sort of span of that event. And so for this study, what we wanted to look into was grassland butterflies. These are often used as indicators of the health of an ecosystem, so they felt like a natural extension of that sort of species diversity monitoring role to be used as a way to monitor genetic diversity. We chose three grassland butterfly species that are relatively common in Sweden, all of them with a least concern status, but most of them declining to some extent in the country. And we selected the common blue, Polyamatus icarus, who is a sort of habitat generalist. You can find it on the sides of roads and sort of very anthropogenic areas, built up urban areas. Then there's the silver-studded blue, which Plebeius argus, which is more of a heathland specialized species. You'll find it on sort of moorlands and in forests under when there's a nice healthy amount of heath in the understory. And then there's the Mazarin blue, Chioniris semiargus, which is a sort of moist meadow specialist. You'll find it in mostly only the more high quality grasslands with a lot of floral richness, and sort of a large area of grassland around it. And this species is one that has seen the most decline of the three, though not yet to be red listed in Sweden. And so we went out and we sampled across this southern Swedish region, different population level samples of these three species where we could locate them in areas that were sort of surrounded by different levels of different predominant land cover types. In general, in southern Sweden, the landscape can be divided into heavily forested areas, which are largely production forests, and heavily farmed areas, which are largely these large-scale monocultures of usually things like oilseed rape and sugar beets. Um, and we gathered these from grassland patches across the region, and got set up. And then we went and compared what we could find in the present day with what was in the museum, and tried to get some comparable historic localities where we could actually get population level samples. And so what is available to us in the museum, of course, is determined by what has already been collected there. And we're very fortunate in Lund to have quite an extensive collection of insect samples, and there's quite a lot of butterflies in there. And thankfully, many of the collectors from the 1930s to 1950s, when we were looking to compare to, were collecting multiple individuals at the same localities within the same year. So we could actually get sort of multiple samples to do actual population genomic estimates from the past and directly compare them to ones in the present. But there's, of course, difference in the sort of representation of the different species. Some collectors collect one more than the other, and they collect in different areas. And there's also often more collections coming from areas with a lot of people living around. Like you can see here, there's a lot more in the southern tip of Sweden for both species, and that's because this is one of the more densely inhabited areas. And so what we could find for all three species was roughly four regions where we could get relatively proximal population level samples to the ones that were present in the modern day. And it sort of went both ways. Some of the ones that were in the museum we couldn't find in the present day because that grassland is gone. And of course, there were many that weren't collected in the past. And so we took these museum samples into the lab. We extract the DNA in a relatively non-destructive way where we use the abdomen and soak it in extraction buffer, but then return it to the specimen, suspended in glycerol. This allows for us to get a lot of DNA, but also preserve the majority of the structures for looking at things like genitalia morphology. Um, and of course, as pretty much everyone who comes to a seminar with this topic would know, is that we get a lot of DNA back that is degraded and damaged. Generally, we found roughly sort of 40 to 100 base pair on average fragment length. But we're very lucky with how fast these butterflies dry. We tend to find very little of this sort of postmortem deaminations, the changes at the ends of the DNA fragments. But even controlling for this and removing transitions, we found in our data that we had a considerable impact from the sample type. So when we performed population structure analysis, including both the modern historical samples, we found that often on the first principal component, we would have historical samples clustering with each other, and modern samples clustering each other, instead of localities clustering with each other, which would suggest that there was more evolution happening over the sort of 50 to 100 years between when we sampled these than there was between these different spatial locations separated by hundreds of kilometers. And we found that the level of gene flow needed for that to be true would lead to us not having this sort of spatial separation that we have on the later components. So we tried to look into what might be driving this and found that it was generally that even without very much DNA damage, we were losing a lot of the alternate alleles in the mapping process. So there was considerable bias towards the reference state in historical samples, and that's expected because of the small fragment size, making it sort of harder for the right context to be found. There's more places that fragment can fit in multiple times, or it might be less confident about allowing an alternate allele in. And while I haven't seen this so much in many other studies having this problem. I talked to a lot of folks at, for instance, ESEB this year who were having the same problem in insects. And it seems to be that because we have in insects sort of almost an order of magnitude more genetic diversity than in a lot of vertebrates, this reference bias can lead to a lot more disparity in the downstream results between these modern historical samples. And so what we did to resolve this was to take our modern samples and call genotypes in those to get a sort of panel of known alternate variants that we were relatively confident in. And we integrated these into the genome using a variation graph approach. And so we could, at the mapping stage, let these short fragments know what alternate alleles they might be able to expect. And this improved the sort of disparity in reference bias between the modern and historical samples. But what we found was that the more heterozygous the species, the less this improved things. So I don't know how limited using this approach will be in the long run. But what we did find was that this sort of reduced a lot of these problems we were having in our downstream analyses, that we were finding now that the spatial structure was the sort of larger explainer variance, and that generally in species where there was stronger spatial structure, historical samples were clustering with their modern counterparts rather than with each other. And so once we had controlled for this, we could more confidently look at the actual genetic diversity metrics we were interested in. And we found that of these three species, we only saw a decline in genetic diversity in one of them, the Keener semiargus, and that's that species that's more specialized and declining to a greater extent in Sweden. And we found that pretty much all of the change in heterozygosity could be explained by increased rates of inbreeding. So it had not yet sort of gone to the point where we were seeing drift eroding away at genome-wide, but mostly that we were seeing that instantaneous loss of heterozygosity from shifting to homozygous genotypes due to an increased rate of sort of background relatedness in the population. And we found that that was coupled with an increase in FST between populations that were at a similar distance, suggesting that there is some level of these populations becoming more isolated in the landscape. And this has led to increased rates of inbreeding and reduced effective population size. And so outside of the neutral genetic diversity, we were also interested to see if we might see changes in functional diversity and if these might have effects on fitness. And we found that although might expect that when populations decline, there could be accumulation or purging of deleterious mutations, we saw little evidence for this at this time scale. And this makes some sense because it takes a considerable amount of time for these processes to start, and these declines are things that have only happened in the past 50 to 100 generations. So we saw mostly that the total count of putatively deleterious alleles was roughly the same between both time periods across all of the mutation categories we looked at and the species that we looked at. But when we looked into sort of how the genotype frequencies at these mutations had changed, we found that the increased rates of inbreeding had increased the homozygosity of these putatively deleterious mutations in at least two categories that we looked at. So we looked at missense mutations, which we infer to be roughly weakly deleterious, loss of function mutations, which we expect to be more strongly deleterious, and mutations at positions that are conserved across this family of butterflies, which we also expect to be relatively strongly deleterious. And we saw that while we didn't see changes in homozygosity in Polymatus icarus or generally in Plebeius argus, except for one category, we saw in Cinereus semiargus in both the missense and the conserved positions, there was an increase in homozygosity that was connected to the inbreeding coefficient. That would suggest this inbreeding is bringing more of these into sort of a realized state. But it is hard to know with these sorts of metrics. We don't have fitness data for them. We don't know necessarily dominance coefficients or selection coefficients. So it's hard to know if this is actually affecting the fitness of the butterflies in the wild currently. All we can say now is that we see this increase in homozygosity that's expected with inbreeding. But if these are roughly additive, it's not going to change the actual fitness expectations for these species. So we don't necessarily have a huge amount of evidence that this decline is actually negatively affecting fitness. But one thing we do want to consider with this is what will happen going forward. So if we look at demographic trajectories over the past 100 years, sorry, I cut off the axis here accidentally, but this x-axis is showing years into the past from the sampling time point. And this is estimating effective population size going back in time using a linkage disequilibrium-based approach. And what we see is generally that these populations saw a considerable decline in effective population size on the order of essentially losing 90% of their effective size in the past 30 or so years, and mostly in the populations in Skåne, the southern region that has had a lot of agricultural development. And this scale of decline is considerably higher than the scale of genetic diversity decline that we see. We've only seen a roughly 6% loss of heterozygosity, but we're seeing almost a 90% reduction in effective population size. And so what we want to consider here is how maintaining this current effective population size would affect the genetic diversity going forward. Will it still erode? Is there some sort of drift debt that is going to continue reducing diversity in these populations? And if we sort of take a simulation along one of these trajectories, we'll do Western Skåne, and then we project it out forward in time, maintaining the current population size, we find that going back from 1950, we see the amount of genetic diversity only really begins declining around 2000. And here in 2020, when we sampled our samples, it's down by about 6% on average in the simulations, which lines up very well with what we measure between the historical and modern samples. And that's this red dot here. But we see that even if the population maintains its current size, it will continue losing the vast majority of the standing genetic diversity over time. And likely, of course, it won't necessarily persist throughout this time, either from stochastic or genetic factors driving it to extinction. But it does help to demonstrate that we are sort of just in the beginning of this decline, and that, for instance, we have a goal of maintaining genetic diversity in wild populations by 2050, but much of that is already set what will be lost going forward if these populations don't increase in size, not just maintain their current sizes. And so what I sort of think we can conclude from this study is that we can see the beginnings of a genetic diversity decline driven by changes in the landscape in Cinereus semiargus, and that it will continue to decline, even if we, say, are able to set aside the landscape in such a way that we can maintain populations as they currently are. And so this would say that preservation of these landscapes alone is not sufficient to maintain genetic diversity in the long term, and for many species to maintain the possibility of long-term persistence. And so when we are considering restoration of nature, or when we are considering conservation, we need to consider that we actually need to do a lot of restoration, and that restoration must be done in a way that maintains gene flow between populations if we are to maintain this genetic diversity in the long term. And now we have in Europe, the EU nature restoration law has gone into force within the past year, but it doesn't make any mentions of genetic diversity or gene flow, rather about looking into sort of the actual spatial connectivity of landscapes. So I think it's important that we continue monitoring and also looking back into what has already occurred in some of these species at a genetic level so that we can make sure that when we are restoring these landscapes, we're restoring them in a way that will enable these species to maintain their diversity long term. And so thank you all for listening. There's been a lot of people involved in this project, both directly and who have helped through considerable conversations, and I want to thank them all. We have a preprint with this of this study out where we'll be revising it a bit some more soon. We've had a lot of very helpful reviews throughout this process that have brought it sort of much better, and so we'll have another revision in the next couple of weeks, but a lot of the technical stuff will still be the same if you want to take a look. But thank you all. And if you have any questions, let me know.
Thanks so much, Zachary, and I'll be sure to post your preprint in the Element server of ARC. Yeah, please put your hand up or just type into the chat if you have any questions. I have a really, oh, Stephanie, you know what, Stephanie, if you're putting your hand up, you can go first then. So go first.
Oh God, you may have already said this, but I couldn't remember. But what was the range of like the individual like butterflies themselves? Like, do they kind of like stray further away, or are they like really isolated in like a very small area?
Yeah, so it varies a bit between species, and we don't have necessarily the best estimates for these because it requires just sort of following a butterfly around. So we have that for some of the species. So one we know with Polymatus icarus, individuals have been observed traveling around 1.5 to 2 kilometers on their own. But generally, we sort of group them into sort of sedentary, intermediate, and dispersing species. And Icarus, the generalist, would be more of a intermediate species. So it's not like migrating across vast distances, but it can move between large distances between patches. Plebeius argus has been the most studied in this. It's one of these species that's good with metapopulation studies. So they've been monitored and shown that most individuals actually don't go more than a few hundred meters in their life, but that a sort of like small percentage of individuals in every colony will always disperse several kilometers to another colony. And Chionius semiargus has sort of the most sedentary of these in that it's not really dispersing very much between patches that are separated by sort of hostile matrix, as they call it. So they generally tend to move along nice quality habitat only.
Thank you. And I guess is that kind of also like a factor in like how I guess well that the populations can react to these things?
Yeah, this makes a lot of sense to me as why it would sort of be affecting semiargus more than the others. A lot of, in terms of genetic diversity, it's been shown that sort of butterflies have very high, um, very, very low local variance effective sizes, but overall have pretty high levels of genetic diversity that are mostly maintained by that gene flow between the populations. So the sooner you cut it off, the more it gets cut off, the faster you get to that sort of local small effective size.
Well, thank you.
Zachary, you might have kind of by way answered my question, which is, do you have any hypotheses why specifically Semiargus was the only species showing that increase in homozygosity? Sorry if you said it during your.
Yeah, no worries. I mean, so there's a few things that I have tried to think about if there's anything special about it. So we've also looked at this not with museum specimens, but with other of these grassland indicator species. So Polymetis icarus and Cinereus semiargus are both on this European indicator list. And that spreads them into sort of widespread and specialist categories. And Cinereus is the specialist and Icarus is widespread. And we've looked at this in more of the species on that list and tended to find that the ones we've looked at that are on the specialist list tend to have these long runs of homozygosity, and they also have these sort of lifestyles of having sort of specific host plants that they need that tend to sometimes be only in sort of very nice grasslands, and they don't tend to disperse as far. So I think it's mainly that this habitat is becoming too fragmented for them. We have sort of regressed the Semiargus's heterozygosity, which is directly related to the inbreeding in this case, with the landscape. And we see that if there's a lot more grassland on the landscape scale, then they have higher heterozygosity. And so it would suggest that it's about the ability to get to those other grassland patches at a broad scale that is mostly uh, affecting it. And so it's that lack of dispersing across this sort of hostile matrix.
And kind of a thanks question. Just coming back to this idea of restoring those kind of habitat, restoring the connectivity between those different habitats. Would you expect the potential genetic recovery to be relatively rapid in like calendar years, just because of their much shorter generation time? Like, is this something that's readily achievable if we
Yeah.
wanted to? Yeah.
Yeah, so I do think so. I think so, like with this sort of lag, this debt and loss of genetic diversity, these species are ones that actually respond a lot faster than others. They have, as you said, short generation times. They have many offspring. So a lot of what has eroded currently has eroded in a way where the actual variation isn't, the common variants haven't been lost yet. They've not drifted away. It's mostly just that heterozygosity is lower because individuals are inbred. And then that just takes a little bit of outcrossing. And this process would occur kind of independently at each patch. So as the patches are connected or even translocations occurred, we would see that they would go up to this historical level. So most of what's been lost by now is mostly the rare variation that would be sort of generated over time. But the common variation is all still there, as far as we can see. We don't see anything that looks like it's been that was previously common that is no longer present in the populations that we sampled.